Hi, welcome to our online lecture, Genes and Tissue Culture Technology. So today I will be going into DNA sequencing and its application. So we'll discuss about the different types of techniques and applications of gene technologies. In particular, we'll look into sequencing. We will compare between first generation sequencing, which is Sanger sequencing, as well as next generation sequencing. So there are multiple platforms available for this next generation sequencing, also known as NGS. So we'll look into the methodology for Sanger sequencing, next generation technologies. There are 5 platforms altogether, as well as the applications, what you can do with this technology for NGS. So Sanger sequencing is the first generation where they uses capillary base and is semi-automated. So I'll give you an example on the shotgun de novo sequencing. We look into the next slide, where the genomic DNA is randomly fragmented and they are ligated with the plasmids. So once it is ligated with the plasmids, and there will be sequence. So all these plasmids with the ligated fragmented genomic DNA will be sequenced and they will be assembled into their original form. So from this assembly, they will be able to derive the genomic sequence of the genomic DNA sample. So the sequencing here, it means that they will determine each individual nucleotide in its original sequence. So how does Sanger sequencing work? So it takes place in a cycle sequencing reaction, and each reaction will involve the incorporation of a fluorescently labeled dideoxynucleotides. So you have heard about what is DNTPs or deoxynucleotide. So what is the meaning of dideoxy? Okay, we'll look into the next slide what it means by dideoxynucleotides. So in this reaction, you have a template DNA strand in which you would want to sequence. So first of all, you will need to have a primer. So for example, this is the primer, and let's say the first letter here is A. So the complementary nucleotide would be T. If let's say a dideoxynucleotide DDTTP is being incorporated, the fragment would be terminated. It will not continue to be sequenced, so it will stop here. If let's say the second reaction, you insert the primer, a DTTP is being incorporated, so the reaction can still continue, and the next one, let's say a DDTTP is being incorporated, then they will stop here. So there will be a series of fragments being generated depending on whether a dideoxynucleotide is being incorporated, then the reaction will be terminated. So if you look at all these variable sizes of these fragments, there will be electrophoresis, which means that they will be separated by electrophoresis. So if you look at this capillary electrophoresis, the smallest fragments would be the one with T, correct? So this is the smallest fragments, that's why they will migrate with the fastest speed. So you have T in the first band here, followed by the second band, which is also a T, okay, and the next one will be G, okay, T, T, G, and it follows. So with the longest fragments derived from G, so you have G as the longest fragment. So if you read the sequence in such a way, from 5' to 3' end, so the sequence of the template strand is the complementary sequence. So for example, here is 3', the complementary sequence here would be 5', G complementary would be C, A complementary would be T. So you would be able to derive the sequence of the template based on these fragments, from the smallest fragments to the largest fragments. So the template for the original sequence would be CTGA. So you can see here is CTGA, exactly the same template strand that you would want to sequence. So this is the example of Sanger sequencing by using 4 fluorescently labeled dideoxynucleotide. So with this technology, they would be able to determine the individual nucleotide sequence by exciting the fluorescent beam for this capillary electrophoresis. Then they would be able to determine the correct nucleotide sequence for the template. So what are the limitations of Sanger sequencing? So if you have observed just now, you will need to carry out electrophoresis. So the discrimination between the fragment sizes is one of the limitations, where they will need to distinguish or differentiate between different fragments that are different by one base pair. So this would be the limitation in the power of discriminating between the different fragment sizes during electrophoresis. And you will need to have sufficient DNA sample, okay, at a high enough concentration before sequencing can proceed. So this is also one of the limitations for Sanger sequencing. So I have a question here. What are the differences between microarray-based technology and sequencing technology in determining genes expression? So you will need to answer your questions in the next slide based on their principle, their background noise, their ability to distinguish different types of isoform, and the reliance on existing genome sequence.